before we get started with Professor Catterall's seminar, a few little things. So the next seminar, I believe, is next week, a week from today, and that's an in-house seminar. And in the spirit of the Cardiac Electricity Center, we try to diversify and have basic science, molecular level talks, as well as clinical talks or clinical translational talks. So the next lecture is really about non-invasive ablation, which is really a whole new technology that is being developed for the future. And here is the little poster for that, and I leave this here for those who are interested. It's a clear collaboration between Cliff Robinson and Phil Kukulich. One is a cardiologist, the other one is a radiation oncologist. It's interesting collaboration, and in our lab for electrocardiographic imaging. So you're all invited. And we'll do this every week. We'll have those little posters for what's coming next week. So that's one thing. The second thing is thank you for being here. And we have a real treat today with Professor William Catterall. I think Bill does not need an introduction, really. And he comes from University of Washington to Washington University. The whole confusion about Washington University. Bill received his undergraduate, I think, in chemistry from Brown, and then went to Hopkins. That I know Hopkins and also chemical physiology or biochemistry or something.
Physiological chemistry.
Physiological chemistry. They have a department like that?
Well, they did that. It's changed names since.
Now they don't call them that anymore. But anyway, he went to Hopkins for that. And then spent a few years at the NIH, NHLBI, doing research, and from there to Seattle, to University of Washington, where he's been chairman of the Pharmacology department for many years. And he just told me that he ended that responsibility recently. So, congratulations. All right, so without further ado, we'll hear about molecular mechanisms of heart failure. Thanks for coming.
Very glad to come back to Washington University, and also especially glad to have my first visit to biomedical engineering and to CBAC. So, we'll talk about calcium channel regulation of calcium channels in the heart, in the fight or flight response, and also how that connects in an interesting way, I think, to heart failure, at least in mice. So, fight or flight response, as I guess you all know, is a conserved response to fear, stress, and exercise among essentially all vertebrates, and probably many invertebrates as well. And it's a dominant physiological response, really takes over the physiology of the animal under those circumstances. In the heart, the key aspect of the fight or flight response that I imagine you all experienced is the rapid and forceful beating of the heart during exercise and stress. So the pathway that generates that regulation has been known for a long time. It's a beta-adrenergic signaling pathway. Begins with activation of the sympathetic nervous system, release of epinephrine from the adrenal medulla and norepinephrine from sympathetic nerve endings in the heart, activation of the beta-adrenergic receptor, adenylate cyclase, generation of cyclic AMP, and activation of cyclic AMP-dependent protein kinase. And these famous folks listed on the bottom of the slide were responsible for working that pathway out in the 1960s, 70s, and 80s. Cyclic AMP-dependent protein kinase then increases the L-type calcium current in the heart. And it's this step that has remained unknown, despite a lot of interest from us and others. And so I'll tell you our progress report on how we think that secret regulatory mechanism takes place. It's more complicated than we thought at the beginning, and probably more complicated than you're thinking. So L-type calcium current, as you know, is critical for the plateau phase of the cardiac action potential. which is initiated by voltage-gated sodium channels, sustained by L-type calcium current. And of course, calcium entry is not important only for the action potential, it generates contraction, and so the force of contraction is controlled to a large extent by calcium entry through the L-type calcium channel. And then cardiac action potential is repolarized by the activation of voltage-gated potassium channels. Very important, but not on our menu today. So a long time ago, when I started in my lab in Seattle, our first major project was to try and understand something about the molecular properties of sodium and calcium channels. A lot was known from an electrophysiological perspective at that time. But the protein molecules that made up ion channels weren't known at all. So first several years, in fact almost a decade, in my lab in Seattle was to try and identify and purify those proteins. And so this cartoon represents our progress. I'll just mention the sodium channel briefly. It was the one we purified first and found that it has a large alpha subunit, which is the pore-forming subunit of the channel, associated in the first preparations with two auxiliary beta subunits. We're not really talking about sodium channels today, so I'm going to jump right over to calcium channels. So we used calcium channel blocking drugs as radiolabels to purify the calcium channel, first from skeletal muscle membranes. They're a very good biochemical source. You'll see we still use them. And the calcium channels from skeletal muscle, or as it turns out, cardiac muscle, have a large alpha-1 subunit that's a member of the same protein family as the sodium channel alpha subunit. And a completely different set of auxiliary subunits. So the auxiliary subunits of these two closely related channel types are really quite different from each other. So jumping ahead then, the slide shows the transmembrane folding diagram of a cardiac calcium channel. I suspect this kind of diagram is familiar to all of you. So these channels, like the sodium channels, have 4 homologous domains. In each domain there is an S1 through S4 segment that serves as a voltage sensor, and an S5 and S6 segment that serves as the pore. But for the story I'm going to tell today, the magnification is on the C-terminal domain, which is truly magnified here. It's not really as big as it looks, but it's a very big piece of protein, 600 amino acid residues just in the C-terminus. So that's as big as most self-respecting proteins all by itself. And it has a number of regulatory sites. And we're going to focus on regulation by cyclic AMP and protein kinase A. And as I'll show you in the next couple of slides, work that's now quite old showed that this regulation requires an A-Kinase Anchoring Protein, or AKAP. We first studied AKAP 15. And that AKAP binds to a site in the distal C-terminal domain here. This acronym, it's an unfortunate one, DCRD, Distal C-terminal regulatory domain. It will come back a few more times. Kinase binds here and must bind there in order to regulate the channel. We've known for some time that the channel is proteolytically processed in vivo in the middle of this C-terminal domain, indicated by the scissors here. And so those two facts were sort of unusual. We're proteolyzing the piece that binds the kinase that we know we need for regulation. So this piece of the channel doesn't get thrown away, it remains, and it binds back to the proximal C-terminal regulatory domain, same unfortunate acronym. And as I'll show you, it becomes a potent autoinhibitor of channel function. And the regulation by pKa takes place by phosphorylation at these sites, which relieve this autoinhibition and give you upregulation. So it's pretty complicated, so I've told you the answer, and now I'm going to show you some data. We'll come back to the same answer in a few minutes. So first we needed to have an AKAP, and we discovered that there's an A-Kinase Anchoring Protein, AKAP, that's physically associated with skeletal muscle calcium channels and also cardiac calcium channels in the rabbit. So we purified and cloned that AKAP. It's called AKAP 15. It's a small protein, 81 residues in length. as a lipid anchor, an amphipathic helix that binds PKA, and importantly, a calcium channel interacting segment here that binds to the same C-terminal site that I showed you, the AKAP binding domain on the C-terminus. It binds through a modified leucine zipper motif. It has 2 leucines at 7 residue intervals. And it interacts with a partner, a sequence of leucine and other hydrophobic residues, again at 7 residue intervals, which is highly conserved among CAV1 family calcium channels. Now I mentioned that the distal C-terminus is proteolytically processed but not thrown away, so this is a cardiac myocyte. And you can see here full length and truncated alpha-1 subunits as we isolate them from cardiac myocytes, mostly proteolytically truncated. The distal C-terminus is here in red, the rest of the channel in green, and they merge with each other and they line up along the Z lines, co-localizing, for example, with alpha-actinic. Right on. So we think then that the C-terminus is proteolytically processed, but retained and bound in at least the same place as the rest of the channel. So the AKAP is required for beta-adrenergic regulation, and that's shown here again from an old experiment from acutely dissociated ventricular myocytes taken from a mouse heart. Maybe it was rat heart in this case, I can't remember. These are calcium currents. Activation gives you an inward calcium current drawn downward here under control conditions, and then with isoproterenol to activate the beta-adrenergic receptor signaling pathway, you get a roughly 2.5 to 3-fold increase in current. You can plot that as a current voltage relationship shown here. If we block the ability of AKAPs to bind to pKa with an inhibitor called HT31, it's a peptide competitive inhibitor, well characterized in John Scott's lab. We perfuse that inside the myocyte from the whole cell voltage clinic pipette. The stimulation is completely gone. So the square sets of symbols here are without and with isoproterenol in the presence of that peptide, and then these are just parallel control data for comparison. So an AKAP is required. Now, do we require AKAP 15 or an AKAP that binds to the calcium channel site? That seems to be the case because here we've taken a peptide that mimics the AKAP 15 interaction site, and again, it completely blocks the regulation. I can't see, is it diamonds? Anyway, whatever the symbols are that went round, show complete loss of regulation if you put in this AKAP 15 peptide. And as a control, we made the AKAP 15 peptide with its leucines mutated so it wouldn't bind. And then it doesn't block, so here you get the same upregulation with the peptide as you do without. So this convinced us that regulation by the pKa pathway requires pKa bound to calcium channels. There's lots of other pKa in the cell, but if you prevent the binding, you don't get the regulation. So it's a key protein-protein interaction in a signaling complex that's needed to get this process going. So the next phase of experiments I'll tell you about, we've tried to reconstitute, if you will, this regulatory system in a non-muscle cell. So you'd like to do that because you have control of all the pieces, right? As long as you study in a cardiac cell, you don't know what the cardiac cell is bringing to the table. But if you do it in a non-muscle cell and you transfect in all the pieces, then you have a chance of knowing what's necessary to get the process going. So we used human embryonic kidney cells, a subclone called TSA-201. And the first key sort of technically important result was that in an HEK cell, the alpha-1 subunit is not cleaved. And so when you express it, you get the full length, which is about 250,000 Daltons, and nothing else. That allows us, in this cell type, to express full length or truncated, and the distal C-terminus as a separate protein. And we can express AKAP, and all the other things you need to make the system go. So the first question is, in this setting, does the calcium channel transmembrane part bind the distal C-terminus expressed as a separate protein. And indeed it does. As you can see here, immunoprecipitation with an antibody specific for the alpha-1 subunit brings down the body of the channel and the distal C-terminus, and that's not seen using antibodies that are not, non-specific IgG as opposed to antibodies. So in fact, these two pieces bind to each other. And that binding has a profound effect on calcium channel activity, as shown here. So these are now whole cell voltage clamped, but with barium as the current carrier. And that means there's no inactivation of the current, or little inactivation, because calcium-dependent inactivation has been blocked. So if we express the full-length channel squares here, we get this amount of current. If we co-express the distal C-terminus, I'm sorry, I said that wrong. No, that's right. We express the full length, you get this amount of current. The next thing I want to say is if you express the truncated channel, you get much more calcium current. So if you take away the distal C-terminus and don't replace it, you get more calcium channel current, more barium. And then if you add back the distal C-terminus, you get this profound regulatory, autoregulatory effect, inhibition of 80 to 90% of the current, depending on how you do the experiment and the day. This is the same effect illustrated as a current voltage relationship. And then on the bottom, the experiment on the bottom of the slide introduces a technique that we use a lot, so I'll take a minute to explain it. So here we're measuring what we call coupling of voltage sensing to channel opening, or coupling ratio. So we stimulate, we depolarize the cell to the reversal potential for the barium current. So there's no barium current here. And we measure the gating current, which is a measure of the charge movement that would activate the channel. So to make a measure of the gating charge movement from one side of the membrane to the other. Also a measure of the number of channels present. And then, after a period of time, we repolarize to the resting potential, which generates this large inward tail current, which we use to measure channel opening. So coupling ratio then is just the ratio of tail current, the current going through the pore, divided by gating charge movement. So it's a measure of the efficiency with which activating the voltage sensor can activate the pore. And you can see that's changed a lot here. This is a situation for truncated channels, very efficient coupling. Control channels, truncated channels, and then channels inhibited by distal C-terminus. You can see the gating charge movement is about the same number of channels, same amount of voltage sensor movement, much less calcium channel current, and then you can quantitate that here in the bar graph. This is a very good measure of channel regulation because you normalize immediately for the number of channels that are present by the gating charge. So with this reconstitution system, we are anxious to find where are the sites of phosphorylation that regulate the channel. And we did a number of different things, but the thing that worked was to look for phosphorylation of sites in vivo, as opposed to taking the purified prep, which we had in sort of biochemical amounts, and phosphorylating it in detergent extract in vitro. All the sites we found by that latter technique didn't have any physiological significance. So to find the sites in vivo, this is now several years ago, and mass spectrometry techniques were not as good then as they are now, we went to the skeletal muscle calcium channel. We purified a lot of it, and you can see a Kumasi glue stain gel here illustrating the pure protein of the skeletal muscle channel, alpha-1S. And then we simply subjected that SDS gel band to proteolysis and then to mass spec analysis, and we tried to account for as much as of the intracellular surface of the channel in the sequence as we could. And of course, the mass spec doesn't just tell you if an amino acid is present, it tells you if a phosphorylated amino acid is present. And to make a long story short, we discovered two sites of phosphorylation that were unknown previously. One at serine 1575 in the skeletal muscle channel, and its mass spec signal for the phosphopeptides is shown here. and another at residue 1579, so 4 residues downstream in the skeletal muscle channel. And importantly, I'll walk over here now so I can see what to point to. Maybe you can't see it either. Importantly, that site is those 2 sites are completely conserved between skeletal and cardiac muscle here and here, as we hoped they might be. And so then we tested cardiac calcium channels to see if they were also a substrate for phosphorylation at these 2 sites. This slide shows one other important bit of skeletal muscle data, and that is to show that these sites are indeed phosphorylated in vivo in the fight or flight response. So here, testing whole rabbits, so to speak, and first extracting calcium channels from skeletal muscle, showing here the immunostaining with an antibody against the skeletal muscle protein, recognizing this large band. And then we add a phosphatase, and of course the protein remains, but now with an antipeptide antibody against phosphorylated residue 1575, we show phosphorylation here, but taken away by the, or staining here, taken away by the phosphatase. So this validates the specificity of the antibody. So then we stimulated the animals by either injection with propranolol, the lowest curve here. This is now measuring heart rate in the rabbits. No injection here, control, or isoproterenol. You can tell we're not that good at handling rabbits because already their heart rate is way up before we inject the isoproterenol. Anyway, if we then look at the skeletal muscle calcium channel protein, it doesn't change during this procedure, but phosphorylation of that site, 1575, changes a lot. So control, much reduced with propranolol and much increased with isoproterenol in the intact rabbit under the conditions of a stress response or a fight or flight response. So that made us even more interested in this site. And now the experiment that I promised prematurely. So we expressed the C-terminal domain of the calcium channel and asked, are these sites phosphorylated in the cardiac calcium channel? Now, the numbers, of course, are different in the cardiac channel, and these are the numbers I want you to remember because that's what we'll talk about from now until the end. So serine 1700 and threonine 1704 in the numbering of the cardiac channel. So these 2 sites, 1700 and 1704, are located right at the interface between the distal C-terminus, which you recall is the autoregulator, and the proximal C-terminus right here. And in the with the C-terminal domain of the channel, we find phosphorylation of serine 1700 in these experiments by pKa and also by calcium calmodulin dependent protein kinase 2, CAM kinase 2. The bottom, threonine 1704, I should have said that this serine is in a classic pKa consensus sequence. Threonine 1704 is in a consensus sequence for casein kinase 2. The phosphorylated threonine is followed by negatively charged residues, 3 glutamates in this case. And you can see in vitro that it's phosphorylated by casein kinase 2, though not as robustly as this site is phosphorylated by pKa. So then we wanted to ask, what's the regulatory significance of these 2 phosphorylation sites, which now we've identified as being phosphorylated in vivo under sort of resting conditions and also after stimulating the rabbits with isoproterenol. So we went to our cell-based assay where we reconstitute channel function with the channel subunits, either truncated or full-length CAVA 1.2 alpha subunit, and AKAP 15, and in this case forskolin to activate pKa signaling. So the left side summarizes these data. So if we start here with delta 1800, so this is the truncated version of the alpha-1 subunit. The proteolytic site is at amino acid 1800. See a large amount of barium current. If we add the distal C-terminus, we get that big intensive effect that I showed you before. If under those conditions you try and activate the channel with forskolin, there are two curves there, and you can see they're not different from each other. There's really no effect of activating pKa signaling under these conditions in these transfected cells. But if we co-transfect AKAP 15 to bind pKa and bind the pKa to the calcium channel, then we restore a pretty substantial amount of forskolin-stimulated barium current, calcium channel activity. So the right side of the slide shows the effect of the double mutation, serine 1700, threonine 1704, which will be called STAA in the subsequent slides. And here, I have to look at it to remember. Yeah, so here's the amount of current with the distal C-terminus and the truncated channel. We increase current if we have AKAP 15 and forskolin present to activate pKa signaling, so from here to here. And then with the double mutation, we reduce not only the basal level, but also the stimulated level of current substantially. Now, this next slide shows the same kind of experiment, but here measuring coupling ratio, so the ratio of tail current to gating current, gating charge movement. And I just call your attention to the right side of the slide. So this is the level of stimulated current after isoproterenol. This is the low level with no kinase, no kinase means a kinase inhibitor present. And then basal level, this is the level of current you would have when you come to the cell. So you can see there's really three levels to think about. Basal, blocking the endogenous kinases and activating pKa signaling. So if we have the complete cocktail, so to speak, plus AKAP 15 and forskolin, we get this high level of coupling, a coupling ratio approaching 30. If we don't stimulate with AKAP 15, or we don't have AKAP 15 present and don't stimulate with forskolin, then we get the basal level, about 9, so about half of the stimulated level in this case. And if we put in this general kinase inhibitor, RO318220, we bring it down to this level of perhaps 7.5 or 8. So that's sort of the parameters. So if we introduce 3 mutations into the channel, serine 1700, which I've described to you, threonine 1704, and a third already known or previously known phosphorylation site, serine 1728. We mutate those 3 sites, we bring the current down to the same level as we do with protein kinase inhibitors. So we think the contribution to the basal current due to endogenous activation of protein kinases is due to phosphorylation of some mixture of these 3 sites. So now we kind of add the sites back one at a time. So if we don't mutate serine 1928, it doesn't do anything at all. So this evidence, plus quite a bit of evidence from other laboratories, argues that serine 1928 isn't doing much. On the other hand, if we add back, that is, we don't mutate threonine 1704, we get up to the basal level, but not the stimulated level. And if we add back serine 1700, we get up to the fully stimulated level. So as the title on the slide says, phosphorylation of serine 1700 and threonine 1704 contribute to the control of basal channel activity. And phosphorylation of serine 1700, which is in a pKa consensus site, controls stimulated activity. So in a way, that's a long run-up of background work that we did at the molecular and cellular levels. And so now we've tested the model that comes out of these cellular level experiments by making mutations in mutant mice. And so that'll be the subject of the rest of the talk, and brings us right up to the current status of this project. So we've studied 3 different mice, but I'm only going to talk about the last 2 in the interest of time, and they're also more informative. So the first mutant mouse, we inserted a stop codon here, and in doing so deleted essentially the entire distal C-terminus. So those mice are very ill. They die of heart failure at birth or even before birth. We took myocytes out from those mice at embryonic day 18 and studied them, and they have completely lost their calcium channel regulation. So that's consistent with the model. But the level of expression of calcium current was also down by about 75%. So the distal C-terminus, or its processing, is necessary to have the normal level of cardiac channels on the cell surface. So this is a complex knockout, but it told us that distal C-terminus is important. If you don't have it, you have heart failure before you're born. And it told us the distal C-terminus is necessary for regulation in intravenous cardiac myocyte dissociated, in this case, from embryonic cells, or embryonic horns. So then the other 2 mouse lines, which I'm going to talk about more or less in parallel, we mutated either serine 1700 to alanine, which we call SA mice, so right here, or both serine 1700 and threonine 1704 to alanine, which we call STAA mice. And they're not identical, but very similar in their physiological phenotype. So I'll discuss them sort of intermixed. So this is kind of a background experiment. We wanted to know that we still had calcium channels in these mice in which we mutated these phosphorylation sites, and that they're located in the right place in a cardiac myocyte. So this slide shows that. So this is a Western blot for calcium channel alpha subunit and the quantitation of many of them. It's not statistically significant, but there's a trend toward a small reduction in the number of calcium channels measured in total. But a better measurement is to use antibodies and confocal microscopy to try and quantitate the channels that are on the cell surface. So we've done that here. You can see the typical images, and these indicate that there's less than a 10% change in the number of calcium channels on the cell surface. And with that preparation, we can also scan across the width of a muscle fiber and ask, are the channels distributed normally? You can see here, if we scan across the width right at the cell surface, more or less the same distribution of channels in wild type, and in this case, SA mice. And if we scan in the middle, again, we see this similar distribution across the width of the myocyte. So we think that cell surface versus intracellular distribution is more or less normal. The distal C-terminus is present at normal levels by, I can't remember now if that's immunocytochemistry or immunoblasts. And serine phosphorylation, pKa phosphorylation at serine 1928 was normal in wild type versus SA mice. So from this, we felt that we have more or less the right number of channels in more or less the right place. They can be phosphorylated by serine 1928. We haven't totally destroyed the myocyte. So let's go forward and do some physiology. So the first experiments we did were on neonatal myocytes. There were 2 reasons for that. First, you get them first, and you're anxious, right? You want to do some experiments. But also, we discovered later that these mice have heart failure as they get older, but they don't have heart failure at perinatal age. So these are myocytes before any disease damage can have occurred to them. So STAA on the top, SA on the bottom. So currents are small at this age, and beta-adrenergic stimulation is relatively modest. So here's the wild type. Without and with isoproterenol varying currents. And then here's the STAA myocytes, lower basal and almost completely lost stimulated effects of isoproterenol. And you can see the same thing here in a current voltage relationship. Same thing for SA myocytes, although it's presented a little bit differently. So this is a time course after breaking into the cell. Presence of isoproterenol right here, and you can see that you get a much larger increase in wild type than you do in SA myocytes. And again, if you look at the current voltage relationship, kind of the same idea, reduced basal current and much reduced stimulated current. So this theme will continue on for a couple more slides. So then we looked at young adults, so about postnatal day 60 to 80, teenage mice. And so these now are current voltage relationships here, with and without isoproterenol at the indicated concentrations. And so very low concentration, 3 nanomolar, you see reduced basal and no stimulated at all. And in wild type, you see a stimulation, but it's not a full-size stimulation by any means. 10 nanomolar, bigger stimulation in wild type. You begin to see some stimulated current in the knockouts, in the STAA myocytes. And then at a saturating concentration for this regulatory pathway, maximum stimulation in wild types and significant but much smaller stimulation in the STAA myocytes. So jumping down to these two panels, sort of an isoproterenol dose-response curve for wild type and STAA. And again, you can see on the axis here that the basal is down, and you can see by looking at the increase that the stimulation is also reduced. You can see that reduction of stimulation a little bit better on the right side. We've just subtracted off the basal levels and looked at the increment in calcium current caused by phosphorylation. Remember, it's the same number of channels, more or less, in both classes of myocytes. And you can see the stimulated current through those channels is much less in the STAA myocytes. And actually, the dose-response curve is shifted about 5-fold to the right, 33 nanomolar versus 7. And almost a mirror image set of experiments. And I won't go through them step by step. I'll just go right here to the last panel where we've compared the SA myocytes, which are in red here, and the STAA myocytes, which are in gray, and their controls, which are located here. Again, this is basal subtracted, so this is the increment in barium current caused by isoproterenol activation. And you can see for both of these mutant mice, it's down about 65% or so from wild type. And again, you can see this change in concentration dependence. The SA myocytes have more or less the same concentration dependence as wild type. There's no change in EC50. But the STAA myocytes are shifted to the right again by about 5-fold. In fact, it's the same data, so it's no surprise that it looks the same. Remember this point, because that's going to turn out to be pathologically important in several slides. So how does that play out at the level of cell contraction? So we looked at single dissociated myocytes and measured their contraction just by video. And you can see isoproterenol enhances their contractile force, as you would expect. And there are inhibitory effects in both the SA and STAA mice that you can see by comparing the black and red. But the effects on contractility are much smaller than the effects on calcium current. And this made us think there's some compensatory mechanism in the signaling cascade that ramps up contraction even though less calcium is coming in. And I'll show you at the end what those compensation mechanisms seem to be.
Do you have any recordings of the calcium transient in this study?
Not really. We don't have anything that is sort of presentable. I'll show you some measurements of SR calcium load.
Because it's interesting whether the regulation comes at the level of the calcium transient or contractility.
Well, I'll show you a little bit of that, okay? But we don't have calcium transient, so that part won't be satisfying in that respect. So then we went to whole animal sort of physiological deficits that we might look at. So this is a measure of the fight or flight response for mice anyway. So here's a little mouse running up this treadmill. And he's running because he doesn't want to fall on these wires down here, which will give him a bit of a shock. It's not a very serious shock, but they work hard to avoid it. So it's really a fight or flight task, and the SA and STAA mice can sustain their effort in this task much less than wild type. They're down 30 to 40%, so pretty significant difference. So exercise capacity is reduced. By P60, we can see evidence of hypertrophy. So if we measure the capacitance of single cells, the SA or STAA myocytes have greater capacitance, so greater cell surface area at the cellular level, and one evidence of hypertrophy. And if we measure the heart weight to body weight ratio, again, we see a significant increase in the SA and STAA mice. So even by 2 to 3 months, they're starting to have evidence of impaired regulation of cardiac function at the kind of whole heart level. So in the last couple of years, we've carried out these studies much longer, and this is a slow process. You'll see this graph goes to 400 days. Maybe it goes to 500 days. You can't do that experiment in a year. And we've found that there's a substantial age-dependent cardiac hypertrophy and kind of a dramatic heart failure in these mice as they grow old. Left side is just showing another controlled experiment. This is a phosphoserine antibody for phosphoserine 1700 you've seen before. It's completely gone in the STAA and SA mice, so we're glad that that's true. It certainly should be true. Also when we measure by immunocytochemistry. So we just follow survival. You can see that the SA mice start about day 300 to die, and they die progressively until they all died by day 400. Our mice live 900 to 1,000 days in normal situations. And the STAA mice are dying sooner. And I asked you to recall that the STAA mutation shifts the dose-response curve for isoproterenol to the right by about a factor of 5. It doesn't change the maximum response. But that shift is sufficient to cost them 100 days of life, so it's pretty substantial. So if you look down below, a heart weight to body weight ratio, you've seen the 2-month data before, or at least similar 2-month data, but by 6 months, even bigger hypertrophy. And this is an example of hearts at essentially time of death in this case, and you can see how large they get just before they expire out here about 300 days. And then panel G on the lower right there is a measure of lung weight per body weight. So this is a somewhat crude, but I think effective measure of congestive heart failure. The lung is bigger because the animals can't perfuse it, and you can see there's about a 2-fold increase in lung weight per body weight. Now, most of these experiments that I'm describing from now on will be on SA mice, because our mouse pills were so large we couldn't do it on both of them. So there's impaired function, as you would guess from this heart failure. There's impaired function when you measure it by echocardiography. I thank Chuck Murray and his colleagues for helping us with this. Doubtfully died, whose name will come up on a subsequent slide. Here, fractional shortening, much reduced in the SA mice, not much effect in the hets. The myocardial performance index, so it's a kind of integrated index of cardiac performance, more or less corrected for changes in smooth muscle function, vascular smooth muscle function. And it is oriented in the opposite direction, that is to say low is good and high is bad, and you can see that there's a major impairment in SA mice. Small change in this measure of ventricle wall elasticity, essentially. While thickness doesn't change very much as measured by the echocardiogram at this age, but if you look in cross sections, you can see that the left ventricle is starting to dilate at this age, and at least in many instances, the wall is thicker than in the wild type. So we wondered whether this form of heart failure, which we've generated now by just taking away one phosphorylation site, in fact, as I sometimes say with a bit of hyperbole, we've only removed one hydroxyl group from the entire mouse proteome. And we've generated this 100% lethal heart failure. So to me, it's a kind of amazing result. So can we exacerbate this form of heart failure by stress and exercise, as one often can in the case of humans with heart failure? So here, wild-type mice treated for 2 weeks by osmotic mini pump with isoproterenol. So this is a mimic of constant stress. You have your beta-adrenergic signaling pathways tuned up for 2 whole weeks. And you can see that there's an increase in hypertrophy that's significant with the isoproterenol in our mice, in the SA mice, but not in the controls. And that's blocked by either carvedilol, a beta blocker often used for treatment in human heart failure. It blocks both beta-1 and beta-2 pathways. And it's blocked by metoprolol, which is important because that's specific for the beta-1 pathway. So we measured running activity, and you can see here 24-hour running activity. And there's essentially no difference in the time course or the extent of running. You can see a comparison of total time running. This is a case where we put the running wheel in a cage and the mice run voluntarily. So it's different from running up the treadmill. This is voluntary running by the mice, but they seem normal in that regard. But if you take the SA mice and you run them, they have an increased hypertrophy, whereas wild-type mice don't. And I'm going to skip the right side of this just in the interest of time. Yeah, that's the slide I wanted. That's the abbreviated form of the data on the previous slide. Sorry about that. There we go. We move on. So this gets in part to the questions that Joran was asking. So we asked, but in a biochemical rather than a physiological way, what are the possible compensatory mechanisms that might be contributing in this disease, and especially contributing to the fact that I mentioned earlier that the loss of calcium current is greater than the loss of contractility of single cells. So the hypothesis was that we've taken away a pKa site. In compensation, the pKa regulatory pathway might be upregulated, hyperactivated, and that's what we found. So these are Western blots here for pKa catalytic subunit, ryanodine receptor, and so on. So let's start with the pKa catalytic subunit. SA mice have more of it by a significant amount. So a lot more pKa than wild type. Ryanodine receptor. Well, there's just the same amount of ryanodine receptor. I don't think we have a bar graph, but there's no change. But phosphorylation of serine 2808, a pKa site that activates the ryanodine receptor, is quite sharply up, as you can see here. So, well, there are a lot of pKa substrates that control contractility. What about some of the others? So down here, you can see that a pKa site in phospholambam is upregulated significantly, whereas an almost adjacent